Good morning, everyone. Today we're going to explore one of the most fundamental concepts in enzymology, the lock and key model. This model was proposed by the German chemist Emil Fischer in 1894, over 130 years ago, and it revolutionized our understanding of how enzymes work. Now, look at this blue spherical structure on the left. This represents the enzyme. Notice the distinctive dark blue cavity or pocket in the center. This is what we call the active site. This is where the magic happens. On the right, you can see this red-colored structure. This is the substrate, the molecule that the enzyme will act upon. See this gray arrow connecting them? This shows the substrate moving toward the enzyme's active site. Now let's read the definition together. The enzyme active site has a rigid, fixed shape that exactly complements the substrate geometry, like a key fitting into a lock. What this means is that just like only the correct key can open a specific lock, only the correct substrate can fit into a specific enzyme's active site. The blue enzyme has a preformed shape, and the red substrate has the perfect complementary shape to fit into it. This foundational concept was proposed by Emil Fischer in 1894, and it remains a cornerstone of biochemistry education today. Excellent. Now that we understand the basic concept, let's walk through the actual mechanism step by step. Step 1. Look at the top left panel. Here we see the red key-shaped substrate. Approaching the blue enzyme, labeled with an E, the enzyme has this distinctive blue keyhole-shaped active site. The substrate is moving toward it, shown by the black arrow. At this stage, they are separate and independent. Step 2. Now move to the top middle panel. Here, the red substrate has fit perfectly into the blue enzyme's active site. This forms what we call the enzyme-substrate complex, written as ES. Notice how the shapes complement each other perfectly. The red key fits exactly into the blue lock. This is the essence of Fischer's model. Step 3. Now look at the large panel. On the right, this is where the chemical reaction occurs. The enzyme, still in blue, now shows the actual catalytic process. See these small colored molecules? We have green circles, orange circles, and purple shapes. These represent the chemical bonds being broken and formed. The double-headed black arrow in the center shows that the reaction is occurring. The substrate is being converted into products. On the right side, we have the chemical equation. E plus S forms a reversible enzyme-substrate complex, which then proceeds to form enzyme plus product. Step 4. Finally, look at the bottom left panel. The reaction is complete. The red key-shaped enzyme is now free again, unchanged. And we see two product molecules, one orange circle labeled P and one green circle labeled P, being released, shown by the black arrows. The text states, enzyme unchanged, ready for reuse. This is crucial. The enzyme emerges from the reaction unchanged and can catalyze another reaction. This is why enzymes are so efficient. One enzyme molecule can process thousands of substrate molecules. Perfect. Now let's look at 3 real-world examples of enzymes that follow the lock and key model. These aren't just theoretical. These are actual enzymes working in your body right now. Example 1. Lactase. On the left, we have lactase, shown in blue. This is the enzyme that breaks down lactose, the sugar found in milk. See the red molecular structure in the center? That's lactose. Notice its distinctive ring shape with multiple O-hydroxyl groups labeled in red. The arrow shows lactose entering the blue enzyme's active site. The enzyme then breaks it down into 2 simpler sugars shown on the right, glucose and galactose. Both have the chemical formula showing CHOH groups and OH groups. This is why people who are lactose intolerant lack this blue enzyme. They can't break down the red lactose molecule. Example 2. Carbonic anhydrase. In the center, we have carbonic anhydrase, shown in teal or turquoise green. This is one of the fastest enzymes known. See this yellow circle labeled Zn2+? That's a zinc ion, a crucial cofactor in the active site. On the left, we have CO2 carbon dioxide, shown as a gray carbon with 2 red oxygens, and H2O water, shown in red and white. These enter the teal enzyme's active site where the zinc ion helps catalyze the reaction. On the right, we get HCO3, minus bicarbonate ion, shown as a gray carbon bonded to 3 red oxygens. This enzyme is critical for transporting CO2 in your blood and maintaining pH balance. Example 3, lysozyme. On the right side, we have lysozyme, shown in orange or golden brown. This is your body's natural antibiotic enzyme. Look at this long chain at the bottom. This is peptidoglycan, the material that makes up bacterial cell walls. Notice the blue hexagonal rings. These are sugar units connected by glycosidic bonds. See these red circles on the chain? The orange lysozyme enzyme binds here and cleaves or cuts these glycosidic bonds, as shown by the red scissors symbol. This destroys the bacterial cell wall, killing the bacteria. Lysozyme is found in your tears, saliva, and mucus. It's part of your innate immune system. Perfect. Now we've reached a critical point in our understanding. This slide compares Fisher's original lock and key model with the more modern Induced Fit model. This comparison will help you understand why science evolved beyond Fisher's original concept. The title reads, Comparison Slide Showing Lock and Key versus Induced Fit Model. We're comparing two competing theories of enzyme action that are separated by 64 years. First, let's examine the left side, the lock and key model, proposed by Fisher in 1894. Look at this brown or tan-colored spherical enzyme structure. It has a smooth, rigid appearance. Inside, you can see the red-colored active site. Notice how it has a fixed, preformed shape. The blue triangular substrate is shown fitting into the red active site. The label clearly states, no conformational change. This is the key limitation. The enzyme remains completely rigid. The brown enzyme doesn't move, doesn't flex, doesn't adjust. It's static. Now, let's move to the right side. The Induced Fit model, proposed by Koshland in 1958. That's 64 years after Fisher. Look at this enzyme structure. It's still brown like the one on the left. But notice these green helical structures wrapping around it. These green spiral coils represent the protein's secondary structure, alpha helices, that can actually move and flex. The active site here is shown in orange color. Why orange? Because it's more flexible and dynamic than the rigid red active site of the lock and key model. We still have the same blue triangular substrate. Now here's the critical difference. Look at these green dashed arrows. These arrows show movement and conformational change. The enzyme is actually changing shape when the substrate binds. See this dashed line outline? This shows the enzyme's original shape before binding. The solid brown shape shows the enzyme after binding. It's actually wrapped around the substrate more tightly. The label states conformational change upon binding. This means the enzyme actively adjusts its shape to better fit the substrate, like a hand putting on a glove. The glove changes shape to accommodate the hand. Now let's examine the comparison table at the bottom. For enzyme flexibility, the lock and key model is rigid, while the Induced Fit model is flexible. The lock and key enzyme is rigid like a brick, whereas the Induced Fit enzyme is flexible like rubber. For the active site property, in the lock and key model, it is fixed and unchanging. In the Induced Fit model, it is adaptive and changes when the substrate binds. For specificity, lock and key is a geometric match based purely on shape. Induced Fit is a dynamic recognition, involving both shape and the enzyme's ability to change. So the fundamental difference is, the lock and key model from 1894 features a brown rigid enzyme and a red fixed active site where the substrate fits perfectly from the start with no movement. It is like a metal key and a metal lock. The Induced Fit model from 1958 features a brown, flexible enzyme with green protein chains and an orange adaptive active site. The substrate induces a shape change. Movement occurs and the enzyme molds to fit. Like a hand in a glove, Koshland's Induced Fit model solved the major limitation of Fisher's model, the rigidity problem. Real enzymes are flexible, dynamic proteins that can adjust their shape to better bind substrates and catalyze reactions. This is why the Induced Fit model is more widely accepted today for most enzymes. This comparison shows the beautiful progression of scientific understanding from Fisher's rigid lock and key in 1894 to Koshland's flexible Induced Fit in 1958. Excellent. Now let's dive deeper into the actual structural details of the enzyme's active site. This slide shows us what's really happening at the molecular level. What you're looking at here is a cross-sectional view of an enzyme's active site. The entire structure is surrounded by this light blue-gray spherical surface. This represents the overall enzyme protein. At the top, notice the label, fixed 3D cavity. This is crucial. The enzyme has a three-dimensional pocket or cavity that is preformed and fixed in shape. It doesn't change or flex. It's rigid and permanent. Now look at this dark blue star-shaped structure in the center. This is labeled as the rigid active site. The dark blue color helps us distinguish this special region from the rest of the enzyme. See all these small colored circles with plus and minus signs? These represent electrostatic charges that are critical for substrate binding. Let me break them down. The red circles with positive charges are positively charged. Amino acid residues in the enzyme's active site. The blue circles with negative charges are negatively charged amino acid residues. The black plus and minus signs on the substrate itself represent the complementary charges on the substrate molecule. Here's the key point. Opposite charges attract. The positive charges on the enzyme attract the negative charges on the substrate, and vice versa. These electrostatic interactions help hold the substrate in the correct position. Along with hydrogen bonds and hydrophobic interactions, these charges create a strong but reversible binding. In the very center, you can see this light gray or white-colored shape. This is the substrate. Notice how it fits perfectly into the dark blue active site cavity. The label on the right says exact, geometric match. This means the substrate's shape is the perfect complement to the active site's shape, like a puzzle piece. There's no extra space, no gaps. It's an exact fit. Finally, at the bottom, we see the most important characteristic of the lock and key model, no conformational change. This means that when the substrate binds, the enzyme does not change its shape. The active site is rigid and fixed. It's already in the perfect shape before the substrate even arrives. The enzyme doesn't flex, doesn't move, doesn't adjust. It's like a rigid lock waiting for the right key. Key features of lock and key model. We'll examine 4 core principles of Fisher's theory. Feature 1, high specificity. The enzyme binds only to an exact complementary shape substrate. In the illustration, we see a beige or tan-colored enzyme with a uniquely shaped cavity. Approaching it is a light blue key-shaped substrate. The arrows emphasize that the enzyme only accepts this exact geometric shape. A differently shaped molecule simply wouldn't fit. This is the essence of specificity. Just like a physical lock only opens with one specific key. Feature 2, rigid. Active site, the enzyme structure, does not change upon binding. Here, the light blue key is now inserted into the beige enzyme's active site. Pay close attention to the light blue translucent outline surrounding the enzyme. This outline represents the enzyme's original shape before binding. The fact that the current structure matches the outline exactly proves that zero structural change has occurred. The enzyme remains completely rigid. It doesn't flex, stretch, or adjust. The active site is preformed and stays exactly the same. Feature 3, non-covalent interactions. These include H-bonds, ionic, and hydrophobic interactions. This is a detailed close-up of the binding interface. The enzyme remains beige, while the substrate is now shown in green to highlight the interaction zone. Look closely at the colored symbols holding them together. The yellow dots represent hydrophobic interactions. The blue wavy lines represent hydrogen bonds, and the red lines with plus and minus signs represent ionic interactions. Together, these non-covalent forces hold the substrate securely in the correct orientation for catalysis without forming permanent chemical bonds. Feature 4, reusability. The enzyme emerges unchanged after the reaction. This is demonstrated in 3 steps. First, the beige enzyme binds the light blue substrate. Next, the reaction occurs, shown by the sparkles where the substrate is chemically transformed into a green product. Finally, the green product is released, and the beige enzyme is shown completely unchanged. The circular recycling arrow symbolizes that the enzyme is immediately free to bind another substrate and repeat the entire process. This reusability is why enzymes are biological catalysts. A single enzyme molecule can process thousands to millions of substrate molecules per second without being consumed or permanently altered. To recap the 4 pillars of the lock and key model, High specificity requires an exact shape match. A rigid active site means the enzyme structure stays fixed. Non-covalent interactions involve temporary forces for reversible binding. And reusability means the enzyme remains unchanged and ready for the next cycle. Now we've reached a pivotal moment in our understanding. This slide outlines the three major limitations of Fisher's lock and key model. While the model beautifully explains enzyme specificity, modern biochemistry has revealed where it falls short. Limitation 1, too rigid. The enzyme cannot account for conformational changes. Fisher's model treats the enzyme as a completely static, unchanging structure. But we now know that proteins are dynamic. Molecules, they constantly vibrate and shift shape. A rigid lock cannot explain how enzymes actually move to lower activation energy or release products efficiently. Real enzymes flex. They aren't frozen metal locks. Limitation 2. No transition state explanation. Enzyme cannot stabilize high-energy intermediates. This is a crucial blind spot. Enzymes don't just bind substrates, they specifically stabilize the transition state, which is the most unstable arrangement of atoms during a reaction. By stabilizing this peak, enzymes lower the activation energy. Fisher's model only shows a perfect fit for the substrate, completely ignoring this critical transition phase. Limitation 3. Limited multi-substrate handling. There is a difficulty in binding multiple different substrates. In reality, many enzymes bind cofactors, coenzymes, or multiple substrates in a specific order. The rigid single keyhole design cannot explain how one enzyme accommodates different partners or adjusts for complex reactions. These limitations led to the Induced Fit model, proposed by Daniel Koshland in 1958. Koshland recognized that enzymes are dynamic, flexible proteins that actively adjust their shape. This modern model solved the gaps by accounting for conformational changes, transition state stabilization, and multi-substrate binding. The lock and key model remains a brilliant foundational concept, but understanding these gaps shows us how science progresses from a rigid lock in 1894 to the dynamic, adaptive enzymes we study today. If you found this deep dive into enzymology helpful, please give this video a thumbs up and share it with your fellow students. Don't forget to subscribe to our channel and hit the bell icon so you never miss our upcoming science lessons. If you have any questions, feel free to drop them in the comments below. Thank you for watching, and we'll see you in the next video.